Hi everyone. So I got the slides, great. So this afternoon I'm going to take you to a travel where in the last 100 years, 4 completely different technologies developed and they merged in the last decade to access and give us access to what we believe is the future of health. So in the next 22 minutes, I swear it's going to be 22, we're going to talk about quantum mechanics, genomics, artificial intelligence, high-performance computing. So buckle up, helmets on. I'm going to try to be as clear as possible, but stop me for any question in the meanwhile. So the story starts about 100 years ago when a young quantum physicist, Erwin Schrödinger, started to think about how would it look a code that designed us. If there is a source code of life, of biological life, what it will look like. So he defined and he gave it an insight that it should be an aperiodic crystal. This was an inspiration for a wonderful team of scientists, Roosevelt and Watson and Crick. They started in 1958 to take a look inside a human cell, crystallize the first biological cell, and see if they could find the code that defined life. Came out the famous X that you find everywhere. That was a picture of the molecule that we were chasing. It was the first picture that we got of DNA. It took us, although 40 years, to manage to poke inside the cell, take out this package of molecules, unroll it, and read it. In the year 2000, a team led by Craig Venter made this achievement. The millennia started being the only species on planet Earth taking a look on the source code of life. We can read it, we own it, and we can read it cheaply today. Now, to get yourself comfortable, you will keep on hearing DNA and DNA all the time. I wanted to take on stage a little bit of it. So this is the code of life. This is a piece of my DNA. And as you might understand, this is my dopamine receptor, as most of you discovered it. So this is a simple alphabet. There are 4 letters. They represent 4 molecules that are stuck together in this helix. And this is what defines who I am, the endowment at the beginning of life that builds up me. So to build a human being, you need about 3 billion of these letters. This is the total size of a human genome. And to explain how many are 3 billion letters, at the beginning of January with Craig, we decided to represent this set of information in a very natural media. The typical access that we have to information is through books and pages. So since it's a code, it's an instruction manual, why not to print it? That's exactly what we did in February. And it's up to be in character 6, 262,274 pages. This is Craig Venter, printed page by page. 175 volume, 1,600 pages each, 3 billion letters. This is the size of a human being, character 6. So this gives you the size on which we are talking about. This is, even though it's a simple alphabet, the complexity is due to a very complex cryptographic system that lasted 4 billion years. Inside those books, there is a history of the solution of nature. There is the ability to grow lungs and breathe. We share most of the code with a long class of animals, down to bacteria. Inside there is the instruction manual that has been proven successful in 4 billion years of evolution. So now, we got it, we can sequence it, we have it cheap, we can collect a huge amount of it. The question is, how the hell we make sense of it? So although it's an instruction manual, and for how proficient you can be assembling Swedish furniture, there's no human being that can follow this book and understand it in principle. So we had 3 techniques to start to understand how the code works. Usually in the last years it's been the lab. The lab, the wet lab, means technically cutting and pasting and seeing the effects, growing small species, understanding how specific parts of the code that we call gene generate some specific function up in the evolution. So the lab has always been and will always be the final proof, but thanks to the fact that we digitalize health, thanks to the fact that we took a molecule and we printed it as a string of letters, this naturally becomes a computational problem. You can take completely different people that have never seen a molecule before, give them the code, and try to ask, can you crack it? Can you crack the code, no matter what it is, if it's a molecule or not? Inside here, there is all the information that you need. So there are 2 approaches that I'm going to show you. They are the bleeding edge of what people are doing now in genomics to understand the code of life. One is artificial intelligence, and the other one is high-performance computing. So we are going to go with artificial intelligence from the top to the bottom, and in high-performance computing from the bottom to the top. AI. So this is a word that you heard, and you will hear in this conference every day. This is the ability to teach and train algorithms or machines to translate computational problem from one domain to another one. Many times incredibly successful. It happens thanks to evolution of a long class of mathematics and algorithms that lasted 40 years, probably 30 years. The idea is fairly simple. On one side, you collect the largest database of genomes that you can touch and that you ever build. The bigger, the better. On the other side, you collect high-quality phenotypes of the species that you want to study. In this case, human longevity, that was the challenge that Peter Diamandis and Craig Venter gave to a team of scientists around 3 years ago. Can we learn? If we have the biggest database of genomes and the biggest database of phenotypes, can we crack part of this code? So the idea is building a machine in the middle. You have several tens of thousands of full genomes, these gigantic strings of 3 billion letters on one side, and on the other, you have every trait that you can get. You have electronic health records, you have phenotypes, so eyes and color of the skin, height, voice, face, anything that you collect of a human being. There's more than 3 terabytes of data of every single human being that characterize one of our subjects, and inside there, we can try to find every correlation that we can get from the genomes up there. So I'm going to show you the history of what happened, because at the end is also a human story, and we decided to start to crack the code with a simple, in a way, approach. So the first thing that we started about a year ago was, can we predict common things? So apart from the rare, which have a complication in statistics, can we predict something very common, like the color of the eye? Can we predict common traits? So we started, can we predict the skin color? Skin color, it's fairly simple. We get to 80% of precision. It's fairly connected to your ethnical composition. Height, can we predict height? We get it fairly good. And height is genomics, let's say the genomic component that explains height is about 80%. So your height is not only defined in your genes. There are also the evolution process, epigenetics. But what are inside your genes? We know it's 80%. We managed to rebuild about 78% of the signal. So we can tell you how tall you are with, let's say, 3 or 4 centimeters of precision. Can we predict the BMI? The BMI is very much connected to your lifestyle. So this is not very much genetic, but still we get in a ballpark. We have an R-square of 0.2. Can we predict your eye color? Eye color, we get it very good. It's 80% of precision on your eye color. Now we step up a little bit the challenge. Can we predict something more complicated? Can we predict something dynamic? Can we predict the age? So the endowment that you start with in your life is not fixed. While you live, the DNA changes. It changes all the time, but the long-term lasting changes sometimes are conserved. Telomeres at the end of the chromosome shrink a little bit, retrotransposon insertions, the code changes. And this is one of the aging, let's say, problems. But can we read, can we understand these differences during the years and we predict your age? Well, we can. And we can tell how old you are with about 6 years of precision. This is something you can do only with full genome sequencing. And then the final challenge that Craig gave us about 6 months ago, 8 months ago, can we predict the human face? So can we find inside those gigantic code, can we rebuild the human face? As well, there's not going to be a perfect model. The face is a very complex trait that comes up with millions and millions of letters in a cooperation. So can we get as close as possible to what we hope for? And that was the challenge. Typically, Craig is a phenomenal leader because he gives you challenges that the industry believes are 10 years away in 10 months in the middle of Christmas. So you really have to speed up to deliver. And what we started, we collected a huge data set. We did the FACE project. What I'm going to show you are the final results. We can discuss technical details later. Anyone that is in data science is more than happy to share details. So this is one of our subjects. So this is a three-dimensional scan of her face. is a reduced complexity in a way. If you can notice, the face has been symmetrized in the genome. There are no asymmetries. Those are collected. And the details of it have been just slightly reduced, okay? The face has moles and facial hairs. They're not in the genomes. You collect them during life. So we reduce slightly the complexity of the face, and we put this as a target. We have a large data set. So we train this machine with thousands of genomes and thousands of faces. We have to be very smart, because the genome is 3 billion letters, but we don't have many billion people. So we have to compress it and find a little bit of a strategy how to manage this amount of data. We get an algorithm that is stored and then learned the conversion. That is an AI, what you call an AI. From that moment on, you can show the AI any new genome, and it will produce a new face. This person has never been seen by the machine before. Her genome has been given to the machine, and a face has been produced. This is the face that the algorithm produces. Now, the wow moment is exactly what I had when I saw it for the first time. I urge you to be honest, to notice the differences. So right now, your eyes are going to right and left to catch the similarities because it's a wow moment. But take a little bit of an analytic point of view and notice the differences. There are many, and it's fair, because it cannot be a perfect model. The nose is a little different, the lips and the eyes, but still, we are in the ballpark. We're predicting a lower complexity, but we're getting there. This is a subject that comes in our lab. This is the prediction of his face. Again, notice the differences. Skin is a little off, the facial structure is a little off, but we are in the ballpark. We are starting to get there, and we're starting to kind of see someone. This is the person that comes in our lab, and this is the prediction of his face. So these results gave us confidence that we were doing things right. So the pipelines were right, the genomes that we collected were high quality, the phenotypes that we got were high quality. It's the very beginning of a result that needs to be improved for many, many years. Many labs are working on similar things, but this is a very simple grasp that will stick in your mind to remember how this process works and how close you can get when you have enough data. Now, you remember we were going from the top to the bottom. We have a lot of data, and we want to deep dive, use the statistics, get down, and identify places or find signal and laws that are embedded in the code. Now we go the reverse, high-performance computing. So the idea is one of the biggest dreams that runs wild since computers have been invented. Can we build a digital representation, a mechanically precise, full, complete simulation of how a human being works, atom by atom, the real deal? Unfortunately, the size is daunting, and we can't get even close right now, but there are sub-problems which are particularly interesting that we can start to tackle. This is a surface of your cells. So what you see is basically a pictorial representation of a very generalistic kind of concept. You have a surface, and you have these strange blobs that grow up on the surface. Those are binding receptors. They're proteins, and they're, in a way or another, the dashboard of your body. Everything that is you, your metabolism, work tapping those buttons. And pharmaceutical industry, at a very essence of it, is managing to find the right molecule that sticks in the right hole. A molecule is a drug. We design it to stick on a specific receptor. When we do it, we can activate or deactivate functions of your body. We can restart your immunity system. We can lower your pressure. This is the pharmaceutical industry. The way we discover binding receptors historically has been in the lab. It's a very complex job. You have to crystallize the protein. It's an incredibly expensive process. But now we know genomics. And we know that we digitalize life. And the idea is we can't represent and simulate the entire human body. But can we tackle this problem? Can we have a digital representation that will help us to identify and do a different class of pharmaceuticals? So we are graduated the last 10 minutes in genomics. So we know that as much as the face, this protein must have a code. And this is the code. So every protein has a piece of DNA, a gene specifically, that encodes its shape. And one of the computational challenges that has been run in the last 20 years is trying to fold, trying to take from this code the shape of the hole that you find on your cells. This is a protein folding class. But there is a step more that you can do, and I'm going to show you a result in a couple of slides that personally I was not hoping to see in my lifetime. So while I was running my team and doing numerical simulation of quantum mechanics at this level, one of the simulations that requires to build up one of these proteins and test if a molecule would stick or not, it was costing about 20 million hours of supercomputer. You had to go to Los Alamos, authorize at the military facility, have access to an enormous football field of computers to run one simulation. So this dream of digitalizing pharma and seeing down there how the atom works has been around since 30 years. But no one had the computational ability to actually make it real. It was so expensive that it was confined to be a simple, a one-shot experiment for very expensive labs. But someone came in rescue of humanity, and nerds. Video gamers. The incredible thing about human beings is that sometimes innovation comes from the most unpredictable place. In the same 30 years where the industry was trying to find a way to digitalize and represent proteins, hordes of gamers wanted to have snappier characters, faster cars, better graphics. And they were so obsessed and the industry grows so fast that they decided to build a complete new architecture, which is called GPU. It's a specific core that instead of solving generalistic problems, it solves a slightly more confined class of mathematical problems in a very fast way. What they used to shoot at characters is a very similar kind of architecture that we need to run computational simulations and quantum mechanics. So nerds came in place, and all of a sudden, in the last 5 years, those simulations that were almost impossible became affordable and became actually cheap. So what I'm going to show you in the next slide is how the pharma industry will look in the next years. We start with a very real example, the HFR and methotrexate. What you get, the HFR, is the whole, is what I want to drug, is my hit in the industry. On the other side is a very famous pharmaceutical, methotrexate. The idea is asking, is methotrexate going to stick in the HFR? I have a biological knowledge that the HFR is what I want to poke. Is it going to stick or not? The HFR has a gene, has a genome, and what I'm going to show you is a very interesting result. So this is the standard genome of the HFR. We call it the wild type. It means the most common one that across the world population is shared. What you see, those letters C, G, and C, those are places where some of us has a slightly different letter. And I report them in the table. There are some of us, instead of having a CG, have a TT. That's totally fine, but it's something that we'll see how it changes things. Some of us has a G where there is a C, some of us has both mutations. Now, this pharmaceutical will work good or not, depending on what letters you have. The binding affinity that I report there in kilocal per mole on the wild type is a good pharmaceutical. If you have the first mutation, you have 10 per less effect. If you have the second, 40 per less. If you have the third couple mutation, the pharmaceutical works 650 times less. You don't know how it works. You don't know which class you are unless you have a genome done. Methotrexate is one of the most standard chemotherapy, and for arthritis is used across the border. Now, quantum mechanics come in place. The results that I show you in the lab took about 10 years. We had to find the people. We had to find the people with specific mutations. This is a quantum mechanical simulation of how the HFR, this big blob, is chewing the small methotrexate. We run it on a video game hardware for 6 hours. Your kid on his Xbox can compete at the top of the level in the pharmaceutical industry. What we get after 6 hours every single mutation, you get the table down there. We can accurately predict at a kilocal per mole energy how a pharmaceutical will work on your specific protein. We take a wild type, a homologous model, we mutate the model, it's all done on a computer, and we show how pharmacogenomics become real, how you react to a specific drug. This is something that was absolutely impossible to think about 10 years ago. It's something that is unfeasible. You will not even write the grant to try all the mutations. This now is done on a video gaming machine. And this is the wrap up in a way. So in the last years, 4 different approaches and 4 different fields grew in an unexpected way, merging together and allowing us to digitalize life and to make simulations and have understanding that we never hoped before. Everything started probably in, let's say, 17, I'm trying to give dates. We can debate on when this field started, but just to give you an idea of how long is this challenge. 1736 is the first time that Carly knows write the term biology in the Bibliotheca Botanica. Quantum mechanics started to be developed in 1925, the Copenhagen interpretation of it. AI starts to be assembled, let's say in 1956, the first Dartmouth conference. And high-performance computing, 1960, Seymour Cray proposes a new architecture to do high-performance parallel computing. These 4 things walked their different paths in the last years. But in the last 5, the acceleration on each of these fields, the ability to sequence genome cheaply, the ability to run quantum mechanical simulation on open source software in a precise way, AI, the ability to extract and manage billion of letters, the ability to scale up and go in GPU computing, these are things that happen literally in the last 5 years. And the things that I always enjoy when I heard the talk and I heard the expression exponential technologies is not really about how the technology shapes, but how many results you can touch thanks to the effect that the technology is cheap and affordable. The results that I showed you today were unthinkable 5 or 10 years ago. So this is the real power of when this technology come together, is the results and the new concept of health that we can build. Thank you.